All right. We're going to
All
let a
right.
few people trickle in here before we get started. Good morning, everyone. All right, P, I think we can get going. People.
Yep, just pulling up the good old slides first. All right, I think we're there. Thanks everyone for being here. Welcome back. Moreover, welcome to our 26th episode of Viewing's You. For those of you that don't know me, I'm Piero Spada. I'll be your host today. I'm also co-founder at Little Warrior Foundation, and more importantly, father now to 15-year-old Little Warrior Lucy. If you're part of our core contingency, aka the Little Warrior Fight Club, thanks for being here with us since day one and making this a place for positive change. And if you're new, well, welcome to Ewing's U. It's our webinar series where we hear from leading sarcoma experts and discuss the cutting edge of care and science behind Ewing sarcoma. Today, we'll be hearing from one prominent expert in Dr. Maggie Fader to discuss the ongoing functional precision medical clinical trial at Nicklaus Children's Hospital in Miami, Florida.
Hi.
Hi, just give me 1 second. Slide change. Here we go. All right, briefly, Dr. Maggie Fader is a pediatric hemonc specializing in refractory pediatric solid tumors and sarcomas. She's the director of the Pediatric Sarcoma Solid Tumors Program at Nicklaus Children's Hospital in Miami, Florida. Her work bridges clinical oncology and translational research with a focus on delivering individualized therapies through functional precision medicine. Dr. Fader earned both her undergraduate and medical degrees from the University of Miami, followed by pediatric residency training at Jackson Memorial Hospital, University of Miami. She completed her fellowship in pediatric hemonc at Memorial Sloan Kettering Cancer Center. Over the past decade, Dr. Fader has focused her research on functional precision oncology. And on the topic of today, she is the principal investigator of a single institution clinical trial that integrates ex vivo drug sensitivity testing and multi-omics profiling to guide real-time patient-specific treatment decisions for children with relapsed and refractory cancers. In just 30 months, the trial has enrolled over 56 pediatric patients and continues to accrue, underscoring its feasibility and clinical value. This work was recently highlighted in Nature Medicine and is part of a growing effort to bring precision medicine from bench to bedside, particularly for patients with limited therapeutic options. Dr. Fader leads or co-leads numerous cooperative group studies, including phase 1 trials through Children's Oncology Group and the Beach Childhood Cancer Research Consortium. She's authored or co-authored multiple peer-reviewed publications and case reports in leading journals. She is also fluent in English and Spanish, and Dr. Fader is also committed to health equity, palliative care, and advocacy. She serves on the medical advisory board for Make-A-Wish Foundation and is on the board of directors for Children's Bereavement Center and volunteers as the camp medical doctor each summer for the annual UOTS Pediatric Cancer Camp. But before we kick things off with Dr. Fader, just a quick reminder that Ewing's U is a webinar production of the Little Warrior Foundation. We are a national childhood cancer research foundation with a laser focus on finding a complete and permanent cure for Ewing sarcoma. We were founded in April of 2020 and have now granted out over $3 million in high potential therapies for increasing survivorship and decreasing toxicity. And in order for us, a Little Warrior, to make programs like this a reality, we rely on the support of the little and big warrior community alike. We appreciate all of your support so that we can continue our mission of bulldozing our way towards more effective and less toxic therapies and continue this vital work for kids battling Ewing sarcoma. With that, I'm going to hand it off to my co-host, Emily, to introduce herself and touch on a couple of housekeeping items.
Hello, everyone. I'm Emily McFadden, one of the other co-founders of Little Warrior Foundation and aunt to Little Warrior Lucy. Today, we will be using the question and answer functionality at the bottom of your Zoom screen. So please send questions anytime they come up through the Q&A platform, and we will address those with Dr. Fader at the end. As a reminder, these webinars are informational in their nature, and we cannot discuss specific cases or provide direct medical advice through this webinar. We're so happy that you guys are all here, and with that, I'm going to turn it over to Dr. Fader.
Thank you both. Thank you for having me here today. Let me share my screen. Hopefully I can get this right. Sec. All right.
There you go.
Okay. So this morning, I want to talk to you all about our research work that we've been doing. Like Pierre just mentioned, it's been over the past decade that we've been doing work like this on a pilot study we had previously, which I'll talk about during the presentation. And now our expanded version of this is underway. So this is the clinical utility and accessibility of functional precision medicine for relapsed or refractory pediatric and adult cancers. Personalized therapeutics for challenging to treat cancers. So I am the principal investigator of this study. This is the clinicaltrials.gov number if you're trying to find it. And I am the director of the sarcoma and solid tumor program in Nicklaus Children's Hospital in Miami, Florida. The objective today will be that we'll discuss the principles of functional precision medicine, and we'll talk about how these technological advances and the expansion of FDA-approved drugs have enabled the bridging of genomics and genomic information with functional drug testing as clinical decision support tools to impact our patients' care. And why is this so important to me? Well, there is a very high failure rate in treating relapsed and refractory tumors. There are very few approved therapies specifically for pediatric and young adult patients. There is the potential with this work to uncover drug sensitivities and combinations of therapy that are new and perhaps have efficacy in treating these tumors. And perhaps functional precision medicine can guide the off-label or investigational therapy use. Just to look at the background and the reason for doing what we do, the survival and outcomes for common pediatric and young adult cancers. So you can now see some of our big ones here, starting with Ewing sarcoma on the left, osteosarcoma, rhabdomyosarcoma, and high-risk neuroblastoma. You can see that in blue, we show the survival, 5-year survival percentage at the time of diagnosis. Ewing sarcoma being about 70%, osteosarcoma close behind, high 60s, rhabdo in the mid 60s, and high-risk neuroblastoma being sadly only about 45% of patients. But then more sadly, look at the orange, and that shows the likelihood of surviving 5 years at the time of relapse. And it's quite low across the board for all of these cancers, meaning that our so-called standard of care options are really not very effective at all. So why are these solid tumors so difficult to cure? There are several reasons. I just highlighted some of them here. There is more therapy resistance at the time of recurrence due to clonal evolution and tumor heterogeneity that builds during the time of the initial treatment and just during the life of a tumor. There is limited effectiveness of our conventional chemotherapies after relapse. There are fewer available targeted or immunotherapy options in pediatrics. They tend to be more aggressive tumors and have a more aggressive biology with rapid progression and recurrence. And there is limited clinical trial availability and drug access for these rare subtypes of tumors. Even when we want to study them, it's difficult to put together a clinical trial because there aren't enough patients to accrue to get data. Some more examples of this. Why is this so complicated? This is just a schemata of the normal cell that actually undergoes some type of insults, be it aging or inflammation, genetic syndrome. And you get this instability, and then a founder tumor cell occurs, which begins to develop. And on the right, in this circle over here, you'll see that after this tumor cell is already developed due to genetic instability, that genetic instability doesn't stop. We continue to get subclones where more mutations are occurring, and we get a very heterogeneous tumor. So some unique challenges in using genomics-only precision medicine for pediatric cancers, meaning we're using only genetic sequencing of the tumor, not necessarily drug sensitivity testing. Pediatric cancers have lower frequencies of genetic mutations than adult cancers do. They have relatively few known driver mutations. So we may find a mutation in a pediatric cancer, but it's not necessarily what's causing the cancer to form. It's more of a signature of the cancer. It's more of a feature that can identify the cancer rather than something that you can target to make it stop. So there are fewer therapeutic options out there for these cancers based on the genomic data. And that's what we continue to find. And if you look at it, we can see that with the genomic data, we look for targeted therapies. And there have been so many breakthroughs. We've discovered target after target, and we've developed all of these drugs. And in the adult literature, you're seeing a lot of efficacy in treating the adult tumors based on targeting these mutations. But in pediatrics, that's not translating. These don't seem to be the answer for everyone. So we've turned to functional precision medicine as our area of research to try to determine whether functional precision medicine guided therapy will yield higher rates of response or disease stabilization than the traditional therapies that we're currently using. What we do is combine the genomic information, do our next generation sequencing with functional drug testing to treat each individual patient. On the left, you'll see that these are the surgeons biopsying a patient with a recurrent rhabdomyosarcoma. They're sending us the biopsy sample, which we receive into our lab and put into culture. We are able to take from that sample both the genomic, get genomic data and get our ex vivo drug sensitivity testing. And with our original pilot, we were able to publish these results in Nature Medicine, and that's a schemata which was featured on the cover showing the physician and their patient with recurrent cancer trying to go through the maze of options out there and determine what's best, looking at genomics, looking at functional drug testing with our artificial intelligence, putting it all together and giving us potential pathways towards better treatment options. The way this works is that we have our patient, in this case, they are pediatric and AYA. Our current protocol goes up to age 30. We take fresh biopsy tissue, whether it's by surgical or by bone marrow aspirate if it's a leukemia patient. We take it to our lab and enrich it for a few days and then are able to run it against up to 150 FDA-approved drugs that are available in our panel for testing. We take the genomic data that we receive back, and the AI program puts all of this together and gives us a personal report with scoring for each one of the agents that we tested. And the whole process takes about 10 days plus or minus. So I would say that our results are usually available between 7 to 14 days on average. Our current protocol that is open, inclusion criteria are patients aged 30 years or younger at the time of enrollment, and they're any gender, race, ethnicity, and you must have suspected or confirmed diagnosis of recurrent or refractory cancer. Patients have to be scheduled for biopsy and have tumor excised fresh. So with solid tumors, that's a fresh biopsy. With liquid tumors like leukemia, that would be a bone marrow aspiration, or if they have them present, peripheral blood sample. Patients have to be willing to do genetic testing, sign consent, and if they're 7 to 17 years old, the child has to assent as well. The only exclusion criteria were whether there was not malignant tissue available and accessible at the time of potential enrollment, or whether what was excised was not sufficient for testing, if it was too small of a sample or if the sample was damaged. We also excluded patients who already had more than a 90% cure rate with safe standard of care therapy. In our pilot study, just to go through what we experienced there, and it was pretty interesting. We enrolled and consented 25 patients. That was our limit. We had one enrollment failure that didn't qualify for the study. We collected samples from 24 patients. 2 of them had insufficient sample given, and 1 had a testing failure. We were able to get ex vivo drug sensitivity testing results on 21 of our patients and genomic data on 20. Unfortunately, 1 patient had rapid progression during the time it took to go through this process, and 1 patient was lost to follow-up because they enrolled somewhere else on another study. We had a tumor board review of the results to determine what treatment plan we would do. 2 of those recommended only surgical intervention and did not receive systemic therapy. 3 continued to have rapid progression before initiating therapy, and we can talk about the various reasons why that happened later on in this talk. And therapeutic intervention was given to 14 patients. 6 of those used the precision medicine guided option, and 8 used physician's choice. There are reasons why physician's choice was used in 8 of the patients, and that's also going to come up in this talk. So the way we do this in the lab is once we collect these tumors, we do a manual dissociation and enzymatic dissociation of the cells from their tumor specimen, and then create a patient-derived culture, which is short-term, 3 to 5 days max, because we don't want more mutations occurring during this time. We want this to represent what's actually present in the patient. We verify the tumor cells are present. You can see that on the upper right. We then expose these into the drug library. We put approximately 1,000 cells per well into the drug library. We can test up to 120 agents at a time, and if we want to run more agents and have enough cells to do it, we will run the additional cells against the remaining drug library. We have quality control measures that verify that we had good specimen present, and we have a drug sensitivity score that's generated. And that measures the IC50, meaning the concentration that was required to get our effect at 50%, versus our Cmax, which is the most you could deliver to a patient in real life, so that we can see whether what we're delivering and is functionally is working in our lab is actually something you could give to a patient safely. And then we generate this scoring system that you see on the bottom right, where this was an example of one of our patients' testing results and the list of drugs with the drug sensitivity score at the bottom, and showing us which drugs were the most effective in the lab setting. So out of our patient-derived cultures, we also looked at what kind of composition is there. There's a lot of physician hesitancy and skepticism on whether this will work, saying that the laboratory is not the human body. There are other factors at play. So we looked at our cultures to say what other cells are present, how closely are we doing in representing what's present in the person. So we saw that immune cell composition was present. We found B cells, CD4 and CD8 positive T cells, neutrophils, macrophages, and dendritic cells, all present in all of our samples. And it just gave some examples of those here. And the tumor purity was analyzed to make sure that our samples really represented the tumor that we were testing. And here we have our results of how patients did. What we measured was the time, progression-free survival, or time to progression of our patients. The top 6 are the patients that received precision medicine guided therapy. And the bottom are those that were by physician's choice, meaning that they did not utilize test results. They used what's considered mainstay treatment options, cytotoxic chemo usually. So you can see on the top of our 6 that used the functional drug testing guided therapy, many of them actually had a complete response, with their time to progression being anywhere from 44 weeks all the way up to 112 weeks at the time that we completed this analysis. We also had a patient with a partial response and a patient with stable disease for a good amount of time. All of these patients came to us with aggressive refractory progressing disease. On the bottom, you can see that 6 out of our 8 patients that did not use the precision-guided medicine approach had progression of disease on the first attempt at delivering therapy. 1 had stable disease for almost 20 weeks, and 1 had a partial response that lasted for about 26 weeks. So we also looked at the power of not only the functional drug testing, but combining that with our genomic information from our next generation sequencing. So our patient 13 was a refractory myeloid leukemia patient. He was 3 when he first presented to us with a FLT3 ITD mutation. In his initial treatment, he took 4 months to achieve remission. He was refractory to four different cycles of therapy before he finally got an MRD of 0. He was treated with standard of care AML therapy, followed by refractory protocol, and had sorafenib as his FLT3 inhibitor along with his chemotherapy. Ultimately, he went into remission after 4 to 5 months of therapy. He had a bone marrow transplant and was in remission for 1 year when he was found to be in recurrence at the age of 5. So at the age of 5, his parents consented to having his blood drawn for testing and enrollment in this trial. He proceeded to get a course of FLAG-IDA, which is fludarabine, cytarabine, and idarubicin. At this point, his anthracycline dose, which is potentially cardiotoxic, was quite high, and he was reaching what we consider the lifetime limit. And we got his drug testing back. His MRD was low, thankfully, with the treatment that he received up front. And we looked at his results specifically to find out whether the idarubicin was really necessary to continue for a second cycle to get him back into remission. What we found looking at our combination results at the bottom was that cytarabine and fludarabine were quite effective on their own, and that the idarubicin only increased the effectiveness marginally. It's the third one down on the bottom right. And so given that there wasn't a big difference and the patient was already close to 1% MRD, we decided to go forward without the additional testing. Without the additional drug, and hopefully saving him from some of that cardiotoxicity. We also saw that the midostaurin as a FLT3 inhibitor was more effective than the sorafenib, so we changed his FLT3 inhibitor accordingly. That patient, slide's missing, sorry, went on to get a bone marrow transplant. He achieved remission in only 2 cycles of therapy. I think it's down on the bottom, number D. Time to complete response in initial frontline therapy took us 4 months, 5 months almost. And he was able to achieve remission in just a month and a half on his relapse therapy using our approach. He underwent bone marrow transplant and stayed on midostaurin for 6 months after transplant, and he remains in remission 3 years later. Our interim analysis of our current study, which is now aiming to enroll and analyze 65 cases of relapsed and refractory pediatric cancer. We were able to show the percentage of samples that were collected by biopsy or tumor resection. The majority are by resection when it was going to occur anyway, but many of them are by biopsy. Sample weight was usually quite small, but we did get a few larger samples. And 95% of our samples were able to successfully be tested and yield results that we could use. A couple of them, we didn't end up getting a sample after enrollment. A couple of them decided to opt on another trial, so we had no request. And one was a drug screen failed due to bacterial contamination. And we've looked so far, this was our first, I think we had 18 patients at the time of this analysis. So we were able to show that we had a complete response in those that are featured in dark green on the right column. And we had a partial response on the light green column on the right, progressive disease at the red at the bottom, and the gray is stable disease. And just to show why this is important and why we look at stable disease as a sign of effectiveness, we have 21 different tumor types enrolled in this trial. The sarcomas are probably our most common request. You can see the spikes there. There's some leukemias at the beginning, Ewing sarcoma right there in the middle at the time of this enrollment, and osteosarcoma and rhabdomyosarcoma are other peaks over there. So the sarcomas are quite difficult to treat and have probably the fewest treatment options of a lot of these tumors. And when you look at how many lines of therapy our patients have had prior to enrollment on this trial, you can see that some of them started directly in relapse, only 1. 2 had 1 line of therapy in relapse prior to enrollment, but some of our patients had 7 lines of therapy prior to enrollment. And that's really difficult to treat at that point because we've already utilized a lot of the agents that are out there that we're used to thinking about, that we're used to treating with, and that have literature and safety data for pediatrics. I just want to go through one case example that kind of exhibits how this actually can make a world of difference for a patient if we find that this is working not only for the patients enrolling on this trial, but going forward, if we find that this is a really valuable tool, we're hoping to prove that endpoint. We had a teen referred to us from Texas with a small round, desmoplastic small round cell tumor of the orbit with a pulmonary relapse. He had received a standard chemotherapy regimen up front with radiation. Then in relapse, he had initiated treatment, but had progression of disease with every regimen they chose. They changed his regimen 6 times and had 2 immunotherapy regimens, immunotherapy drugs included in those 6 regimens. He had no response to therapy. The patient was enrolled in hospice, and he'd been sent home with methadone for refractory pain. When his physician heard about this study and contacted us asking if he could enroll the patient in the functional precision medicine study. We agreed, and the patient was determined to be too ill to travel to Miami for enrollment, so we were able to facilitate enrollment remotely. We shipped the media to their site and gave them instructions on collection, and a biopsy was taken. We verified tumor cells were present. They arrived intact. Everything went well, and we were able to generate results for this patient. These were the top choices. He actually did not have a ton of medications show up as effective on the testing, but of those that did show effective, carboplatin and trabectedin both showed up. These are not medications we traditionally think of with desmoplastic small round cell tumor, and we don't think of them often with a lot of our sarcomas. So limited data on utilizing them, but our physician decided to go ahead and do that. And so just to compare, we had the physician's choices, which were not directed by any precision medicine data. And he'd gone through 6 lines of therapy, 2 immunotherapy lines, and had progression of disease. Then we enrolled him on the trial, utilized trabectedin and carboplatin, and we were able to get great results. And the physician wrote us this very nice letter to express his and the patient's gratitude for this testing. But basically, after all of that this child had gone through, 2 months after treating him based on the findings, he was off the methadone. He was back in school full time, back into his activities with his friends, and had his first treatment response since relapse. And they sent us some of the radiographs and CT imaging so you could appreciate it. But we have left prior to enrollment and right after 2 months of therapy from March to April on the top left. And then on the right, you can see the CT data showing from March to August how much improvement he's had, almost complete resolution on the right hemithorax and quite a reduction on the left hemithorax with minimal disease compared to where we started. And the child continues to do well to this date. So we're awaiting an update on his fall imaging, and hopefully he's still doing well. Some of the challenges that we've met along the way, when we originally started this trial with the pilot 9 years ago, we had very slow enrollment and only accrued 5 patients per year. And that's pretty impressive because, as we know, we just saw in the data, 30% of those patients have a recurrence in 5 years, even with localized disease for the majority of those main tumors that we're treating, including Ewing sarcoma. So what were the problems? Well, originally there was a lot of physician hesitancy. There's skepticism. Does this really work? This seems too easy. I don't know about this. And a lot of people like the more structured clinical trials where you're testing a specific drug in a specific way with every patient, and they all meet the same eligibility requirements. And it's what we're used to. It's the tradition of clinical trials. So with increased physician familiarity with these new approaches in oncology and with personalized medicine approaches with genomics, I think that started to influence clinical decision-making and the referral patterns. There's the need for the medical community to collaborate more with this type of research and the new technologies so that we can make better informed treatment decisions for the patients. So I think that has changed, and we have seen a huge uptick in the referral. I think people have started to realize that although you may have skepticism about trying out a new method and maybe repurposing different drugs that weren't originally indicated for the specific disease, I think people have come to realize that relapse cancer outcomes are so poor that it doesn't make sense not to try something new. It doesn't make sense to offer patients the same therapeutic options that only give them a 12% chance at 5-year survival. So I think we're having people being a little bit more open-minded about investigating this now, and we're getting more enrollments. Sometimes we have barriers with the willingness of the treating team to biopsy or the difficulty in biopsying a site. So sometimes that's a problem. A huge barrier we have, and it's why some of the patients in the original pilot did not get the precision medicine guided recommended option, but physician choice, FDA approval and insurance reimbursement. The access to off-label use of drugs is creating huge financial restrictions for our patients and families. And many times when we do identify a preferred treatment protocol, we can't get it for them. And so some of these drugs are extremely expensive. We could be talking about $30,000 or $40,000 per month to be able to get some of these, especially oral agents that are new. And that's been a big barrier for us, trying to implement the therapies that we're finding. and ultimately demonstrating the reliability of functional drug testing. So I think that our pathway forward is that we want to enroll enough patients and gather enough evidence, hopefully, that we can show that precision guided medicine options do lead to better outcomes and more chance of response, more chance of stable disease, and hopefully more chance of cure, that it becomes an approved tool that's covered by insurance, just like NGS. If we went back 10, 15 years ago, next generation sequencing was not being routinely covered by insurance, but now it is. And we hope that this becomes another tool that is able to be accessed by any patient. So our future directions are that we are intending to develop a consortium platform to further evaluate and validate the findings from this small feasibility study. What we have now is still considered a feasibility study, and we're identifying our barriers. Through a consortium, the biopsy and collection procedures will be feasible for more children. We can develop a regularly scheduled tumor board between the hospitals participating to review both the NGS and DST results together and decide on a treatment plan with each enrollment and do follow-ups on these patients. Development, we hope we can develop subsequent clinical trials for specific disease types, which incorporate our functional drug testing findings and tumor types. So for example, if drug X is seen consistently for soft tissue sarcomas in vitro in our lab, then maybe that drug X warrants a formal clinical trial and ultimately FDA approval. And we also hope to get the opportunity to publish a lot more of these cases to establish more literature for the off-label use of drugs in various pediatric cancers, which is really important for us in getting those drugs approved by insurances and getting those drugs to be accessible to the patients that need them. And I just want to end with some of my kids who are our why we do this. And thank you for listening.
Wow, thank you for that amazing presentation and for what you're doing. As I'm looking at the Q&A, I see one rabble-rouser in the group who just said, repeated your quote back. It doesn't make sense not to try. And so that is really powerful, and thank you for trying for these kids. Can you stop sharing your screen? Because I can see myself
I'm still
twice, and for some reason it's flipped.
Yeah.
Oh, there, it's up there. Sorry.
A different
I thought I
version
had
of myself. No, thank you. I'm easily
I
distracted.
got out of the presentation, but I didn't hit the stop.
Yes, no, thank you, thank you. So we have a couple questions about this trial being located in Miami. Does someone need to travel to Miami to get their biopsy? Do they need to be treated in Miami once they have a protocol selected? Could you talk a little bit more about the local
Yeah.
versus
Yeah,
remote options?
it's been an evolution, and I think that was part of the feasibility that we learn as we go with this type of research, because this research is new, and we're doing stuff that's kind of off the cuff. It's not traditional. When we first wrote this, the grant funding was through the Office of Minority Health at the Florida International University. Why? Because based on our demographic of South Florida, we had the majority of our patients down here happen to fall in minority categories. That's not the case anymore after our administration kind of dropped this and we had to reapply and kind of change the trial around a little bit. So right now, what we have learned as we've gone is that once the trial got recognized and we started getting a lot more inquiries, we got inquiries from around the country, not just from South Florida. And as we did that, we ran into, of course, what comes with that: financial barriers of travel, medical barriers to travel, patients maybe not well enough, and then Medicaid barriers to travel. If all the state insurances do not easily allow people to go, and these are time-sensitive issues. So we made an amendment to the protocol to allow collection remotely. And we made a pathway for that where we ship our own media to the site, send the instructions to the site with the collaboration of the site physician, and have it overnighted back to us. And we also created another amendment to allow for a remote consent process. So the patient can consent to everything by telephone consent and not have to come to Miami physically. So I think that's allowed us to enroll like this patient that I just presented out in Dell Children's. They never came to us. I haven't treated this family. The only thing that we've had contact with is their physician and with them for enrollment. But everything else, we're collaborating with their physician. And if the family wants to speak with us, we also speak with them as well. But in this case, it went through their physician, who's been great with working with us. And he's actually sent us 4 or 5 kids already who we've not a single one has actually traveled to Miami. They've all been collected remotely. We just do the testing, talk about the results, kind of help them figure out how to use them. And then we ask them to send us the data back for what they decided to treat with and follow-up imaging results or testing results so that we know whether or not this is helping. We're able to measure the outcome. And that's what we're hoping we'll move to with a consortium trial that will have more hospitals that are kind of better staggered across the country so that this becomes a little bit easier.
Are you guys affiliated with a consortium already, or?
We're affiliated with a couple. We're kind of debating whether we want to go through an existing one or whether we want to just make our own, for this study and partner with just a
Mhm.
handful of hospitals. So part of that depends. We have to get funding for this. So that's always the yay. And I think we're a little shy about getting government funding at this time for many reasons. So many things have been cut lately that we want to have sort of a reliable source that we're not going to worry about politics getting in the way. And so, yeah, I think our next step is getting the funding. We've identified a handful of hospitals that are extremely interested, some of whom have already been working with us and have already been sending us patients.
Mhm.
And we may identify a couple of others too. So, I mean, we're going to talk about how many patients we want to accrue, but we're thinking somewhere between 200 to 300 patients and maybe 6 to 8 hospitals.
Nice. I just want to build off of that, and I kind of like want to hone in on this point, Dr. Fader. First and foremost, thanks for everything you're doing. It's obvious that you have the patient, front and center in terms of like urgency, right? Like being able to get a biopsy remotely, creating a clinical actionable plan in less than 10 days. That's phenomenal. Can you kind of like just highlight that really the functional precision medicine plan that you prescribe is really treatment doesn't need to happen at Miami, right? It's all about like fostering a medical alliance with wherever the patient's being treated.
Right.
Is that correct?
There's a few things about that. So one thing about that is with the remote collection, one of our barriers has been how well we can manage it at the treating institution and how much the treating team feels comfortable with doing this. Some of them have been like, let's go, we got this, and some of them not so much. And that I think is something that might be helped with this consortium platform because if their home institution can't do it, maybe one nearby can, and they don't
Mhm.
have to travel from California to Miami, let's say, or something that's really difficult. We had one come from Wisconsin that's a long way. So maybe there's one in Cleveland or maybe there's one in, the Southwest somewhere. So something closer to home is what we're trying to help get there so that there can be a home base that's nearby if we can't do it with their treating institution. And then no, the treatment doesn't have to happen with us. The only time that that's happened is when the home team hasn't felt comfortable using those results. And so far, we haven't really had that as a barrier. I think only one patient has come to us for that reason. Okay, no, two. Two patients have come to us for that reason that live not too far from us. And so I think that we would really like to have good collaboration with the home team that's taking care of the patient and keep them from having to travel and try to keep the burden low because it's already big enough as it is. Um, and then the next barrier is like the access to the treatments. Is the institution at home able to get these treatments? Are we able to establish a treatment plan that will be covered because they can be super expensive? I mean, we even tried to get a drug fund we were fundraising to have a drug fund for some uncovered drugs. And even then it's of limited help. I mean, it does help if you have an IV medication that maybe costs $7,000 and we're not able to get insurance approval and we say, okay, we'll use the money from the drug fund to buy the first dose. But eventually we need to either get approval or something else has got to give because that's not going to suffice for a whole treatment plan.
Thank you for that.
Right.
Do you think, I
Oh,
guess,
sorry.
building
One
off of
other
that?
thing I wanted to say. I'm sorry, I almost forgot.
No, sorry.
The other thing about this is that it's a unique trial and then it doesn't obligate you to a certain treatment protocol. You get the results and then the family and team can talk to each other about what kind of treatment they want. Do you want to just go to the highest number on there? Do you want to think about oral medications versus IV medications because you don't want to be in the hospital? Do you think about meds that aren't going to make her lose her hair? There's not a specific mandated treatment for any one patient. It's workable based on your goals and based on what you want. And there's also a lot of thought that has to go into it about once you get those results, how can you use them safely? You can't just grab the first 5 drugs on there and say, we're going to use these. Those might not work together. Those might not be safe together. Those might even be antagonistic in some cases. So, a lot of work goes into that from our end on trying to make sure that what we're putting together from those results is actually feasible in a safe way. So it gets complicated.
So that was going to be one of my questions, like how do you put that together, right? Since it is so personalized, and like say you got 5 drugs that are recommended for individual patient, like how do you piece that together to be like, well, only 3 of these are tolerable, or like how does that work?
Yeah, we have limited pharmacy support right now. We had a pharmacist on there, but we're kind of in between right now based on funding issues. But I think going forward, that needs to be a critical part of the platform, that we're going to have pharmacy support so that someone there can help us guide. Right now, that's a lot of me doing that. But researching the peak, the half-lives and the peaks and what drugs are going to be antagonistic or dangerous to use together, because sometimes they don't allow each other to clear, and so you get toxic levels of the drugs if you put them together, or they have the same adverse effect profile, so that's too much in that realm. Like, let's say 2 different drugs both have a high rate of causing neuropathy, then we don't really want to use those together. Or they both use the same cytochrome in the liver to be excreted, don't want to use those together. So it does get a little complicated, and that's where I think it's important to have a lot of centers that are working together on this.
Thanks. Go ahead.
How much tumor sample is needed? Is there a size limit or a mass limit?
No, it's more like how cellular it is. That's like the $1,000,000 question, and I think it changes with every single patient. So we always just say as much as you can give us. With the bone marrow for a leukemia patient that's in relapse, it's really probably not that much. Maybe somewhere between 5 and 10 mLs from a bone marrow aspirate is probably fine, and that might even be more than enough. It just depends on how cellular it is. When you're looking at tumor specimen, you have to remember that not every piece that you get out is cellular. Sometimes it's got a necrotic or bloody area in it, or sometimes a partially treated area. If that patient's had some chemo before or some radiation before, it might be damaged. So we never know what we're going to get. So we used to say, 2 to 3 grams of tumor. Some people go, that's impossible. And sometimes just a little core or 2 is enough. I recently did a bone marrow biopsy from a neuroblastoma patient of mine, and 2 cores was able to get us enough cells to test 70 drugs out of the library. So it was very cellular. I mean, 2 tiny, tiny cores. So sometimes that's enough. Sometimes it's not. We just say give as much as you can.
Awesome. Switching gears here, one of the slides that jumped out the screen to me was in the pilot study when you were presenting the clinical outcomes of the precision medicine versus physician's choice. It almost seemed like black or white. Like why wouldn't you choose the physician's choice, or excuse me, the functional precision medicine versus the physician's choice? So there's a question here from one of your colleagues that says, he's looking at the figures, and it says progression-free survival was less than a week in the physician's choice, or presumably talking about IT, cyclotopo, et cetera. It is unlikely that they received sufficient treatment to know if there was an effect. How should this be interpreted? I worry about selection bias influencing results. Do you have any comments there on that initial pilot study and that control group, so to speak?
They were all people that progressed during the first cycle of therapy. So
Okay.
they were, in cycle 1 when they showed signs of progression, didn't even make it to cycle 2.
Okay.
As far as physician choice and biopsy, so a lot of those did want to go. So this is where I go back to the access. Many of those patients, it wasn't that the physician, I shouldn't say choice. That's probably not a good way to phrase it, but the easiest therapy accessible would probably be a better way to phrase it. Sometimes we wanted to use one of the precision medicine guided options, or one of the several options that we had there, but we couldn't get it. So they used what they could, and what they could get an insurance approval for that would be considered an acceptable regimen to the insurance company for that indication.
Thank you.
There's a question here about AI. How are you using AI in this trial and analysis?
The AI, we have a program that we use to put in the data from the drug testing results that creates a scoring system based on it looks at the Cmax and the IC50, and the amount of cells that died during the assay, like amount of cell killing during the assay. And then it generates a scoring system. It gives you a drug sensitivity score. And from that, it will note that if it had a really good, so sometimes the drug has a great effectiveness in the lab, but at a concentration that was higher than what we normally give, higher than our Cmax. And so that's noted. If our IC50 is higher than our Cmax, there's a line indicating that although it was a very effective drug, it wasn't effective at the IC50 was not at a concentration that we normally give to people. And so we might say that although it got a high score, we wouldn't use it clinically. So that's a program that helps us make that calculation.
Very cool.
We got like 10 questions here, so we're kind of going to rapid fire if that's all right with you, Dr. Fader. Going all over the place. This one
I'll do my best to answer.
Patient expense. I don't know if they misinterpreted this, but it says there is no expense, right?
No expense, no.
Okay. But they have
Well,
to
the biopsy.
travel.
So a biopsy is done as a standard of care, so it's not an expense to us. We get nothing, but whoever's doing the biopsy is going to do it as a standard of care. It's the biopsy itself is not part of the study because we just take the tissue from a biopsy. The idea is that you're getting a biopsy, either you're getting a resection of a tumor, or you're getting a biopsy to confirm a relapse, or you're getting a biopsy because you want to send next generation sequencing or something like that, and we're taking additional tissue. So the biopsy is considered a standard of care that should be hopefully covered by insurance at the treating institution, wherever that is. And your physician care, once you get on treatment, is your standard of care. Whatever I do is not charged, and whatever the testing is not charged. So all of that is part of the study.
And what about the treatment? That's totally up to the insurance.
Totally up to the insurance or wherever they can get it from.
Okay, thank you.
Can a patient self-refer?
Yeah, they can reach out to us. We just need their team to cooperate with us as far as getting the records and data and things like that. And somebody's got to we got to find a place to do a biopsy, whether it be with us or at their institution or somewhere else.
All right, there's lots of people on here singing your praise, and then asking, are you aware of groups in Europe, or is there plans to take this outside of the US, etc.?
I'm not aware of groups doing this in Europe. I think there's places around the world doing xenograft models and some other types of personalized medicine models like this. The xenografts take a lot longer. This is like the turnaround time is more efficient. And then the xenografts, you worry about them like continuing to mutate as they're growing over that period of time. So there's like pros and cons of both. We have had requests from Europe, and we're scratching our heads trying to see if we can do it. We've had one patient sent from Canada, and we have another one waiting, and another one that's inquired from Europe. And we're actively trying to see if we can coordinate that, where we can get them the media and get through customs in a timely fashion that the cells don't die. So we're looking into seeing what we can do.
Great. A patient here says, oh, what is the first step for my son to do this? How soon can we start?
So first step is just reach out. We actually have an email. It's ex vivo. Wait, tell you exactly. Hold on. We have a clinical trial email that you can reach out to, and I'll get all of our coordinators on board right away. Let me tell you what it is so I don't give you the wrong. Yeah, so exvivotrial@nicklaushealth.org. So EXVIVOTRIAL at Nicklaus, which is NICKLAUS health.org. And if they email that with their information and contact, then somebody will get back to them within a couple of days. Usually same day, but just to be fair, just in case, give it a couple days.
Okay. I got kind of a bigger picture question here. Like, what is the end goal of the study? What is your end goal for this in totality? And is this ever going to make its way to frontline, do you think?
Yeah, so the big picture for me is that I want to continue collecting data as long as it takes for us to publish a big enough series of patients. And hopefully this continues to be successful because, like I said, this is a pilot feasibility study we have published. We have to continue to replicate that data and hopefully it continues to work. And it's also hard because we allowed patients I mean, there's no Lansky, there's no eligibility that requires a performance score. And perhaps we need to put that in. You know, I hate to ever say that we don't take everybody, but it factors into what you can realistically hope for. Like we have several patients that, as you saw in the pilot, had rapid progression of disease while waiting on results because they were so advanced when they got to us. And so maybe that's not a good idea to accept that patient for that trial because it's just too long. It's not practical. But I think the big picture is to get enough data with enough children that have successful outcomes to be able to get this to be an approved tool that can be utilized in any scenario. So first you test it in the relapse refractory setting because that's where you have the biggest need and where you can make the most difference. And then you maybe test this in frontline. For example, if I have an osteosarcoma patient that's already coming to me metastatic and I already know that the chance of cure for both Ewing's and osteosarcoma are 20 to 30%, 5-year survival at diagnosis because of the metastatic disease, then maybe this should be used frontline. You know, maybe this is something where you can add in additional therapy options. We're also looking into starting a trial where we're going to this particular one, it wasn't covered, and we've done it off label for some people. But we're looking at a clinical trial that'll also show repurposed drugs that are not necessarily cancer drugs. They're not labeled as such. They're not
Mhm.
anti-neoplastic, but maybe everybody's hot in the media right now. Everybody under the sun wants to know about ivermectin and fenbendazole and mebendazole, and I could give high dose vitamin C, high dose vitamin D, you name it. Okay, so we'll test them. And I would say I would love to give you, you know, vitamin whatever if it works. I mean, I don't get paid
Mhm.
by these drug companies for giving you anything. I don't want to prescribe a $40,000 drug. I get super excited when the drug sensitivity score actually shows me that irinotecan and entamidar is really effective. I'm like, yes, because that's easy to
Yeah.
get and it's cheap.
Right.
And we can treat the patient easily and quickly, and that's fantastic. But when it shows me that none of the real stuff works and we got to go to these crazy fancy drugs, then it's like, ugh, like, how are we going to use
Yeah.
this together? How am I going to get that paid for? So clearly, if something that was, honey in your backyard worked, I would love to use it. We need to test it. So, and we need to be smart about it. So we're talking about doing a small pilot study on repurposed drugs and a repurposed drug library to see if that also can add into this.
So really what you're looking for approval then is the method that, right? Like it's not really
Right, that you
about
show a pattern
approval.
right, when you use this testing, just like next generation sequencing identifies, so
Yeah.
you used to have to pay $5,000 a panel for it. You got your results, and maybe 5 or 10% at most had an actionable target, but it was enough to say that still matters for those 5 to 10% of patients to make this
Hmm.
an approved drug that approved panel that insurance pays for. So you wanna make it so that people say, "Yeah, everybody should have access to this tool, and we use it, we send the sample, we process it just like NGS, and you can use that in your decision making for your patient." And we know that not all cancers are the same. They're all mutants. They don't all respond to the same therapy. If they all responded to the same therapy, they'd all have the same outcome. So I think that that's the argument for saying that there's got to be some personalization to each kid's approach.
Well said. Any other questions here that you want to toggle off?
There's one here about, do you support the application to a drug manufacturer for compassionate use when the drug isn't covered? Is your team assisting with
Yeah,
that, or how does
we try all the time. We have our research department helps us with it. A lot of companies don't offer it, so that's a problem. When they have, we have applied, and it's not a fast process. It usually takes us 4 to 6 weeks minimum, even when we're hounding them every day. I mean, I've had where I'm chasing them down, and it still took some time. We've been successful sometimes. So if they have the program, we try for it.
Mhm.
I actually reached out to one the other day and I got a hard no. So uh, yeah.
Hmm.
Do you see any other questions on here? I don't have any other questions. I see some patient-specific questions, but that's maybe for another time.
Now, if on the line of the accessibility of the drugs, if this tool were validated to show that it does actually makes sense to use it and it does give you better options for therapy, then maybe that would then mean that if the tool points to whatever drug X is, then the insurance needs to pay for drug X. That's your tool. So they like to say, well, where's your phase 3 clinical trial that shows the evidence? Where's your, whatever, they want a phase 3 clinical trial or multiple phase 2s or some evidence that this has worked in this cancer type before. So if this tool became validated and you said this type of testing actually does tell you which therapies are better, then maybe that becomes the new standard for Medicaid to say, yes, we approve the drug that's indicated by the testing.
That makes sense. I guess my last question on that, is there enough overlap? Or are these each case so individualized that it's never going to just be like this combination needs to move forward?
Well, I'm personally seeing overlap in a lot of soft tissue sarcomas. So
Okay.
not necessarily only in Ewing's or only in rhabdo, but I've seen that there is a difference in what I'm finding between recurrent refractory osteosarcoma and Ewing sarcoma and how well they respond to some of the drugs. Sarcomas in general, I'm seeing signals of particular types like HDAC inhibitors and proteasome inhibitors coming up very frequently, and a lot of repeats of the drugs that are coming up. So that's why I said a secondary aim would be that if we really see that these are coming up a lot, and then we see them actually being useful frequently, maybe it warrants its own individual trial so that that could become an actual FDA-approved drug, one less drug to fight for.
Got it. Makes sense. All right. We're close to the end here. Emily, do we have any other questions in the Q&A box that we need to get answered?
I haven't seen anything fresh come through.
Okay. If not, we'll bring it to a close. Thank you so much, Dr. Fader. This is truly a revolutionary clinical trial that you're running. Thank you for your grit, your gumption, and your urgency, right? You think like a patient, and from the patient community, thank you so much. Not only for kids, diagnosed with Ewing sarcoma, but all refractory relapse kids that are fighting cancer. So thank you for being here and sharing this clinical trial with us all. I want to thank my fabulous co-host and co-founder, Emily, as always, for helping me moderate conversation today. And lastly, thank you to our viewers for joining us today. Please remember to stay up to date and subscribe to all our Ewing's U emails. You can do that by going to littlewarrior.org, navigate to the Ewing's U tab on our website. You can also follow us on Instagram and Facebook, as we always post about our next speaker that way as well. That's it for now. Hopefully you enjoyed this episode. We'll see you next time in true Little Warrior fashion. Swords up, everybody.
Swords up, everyone.
All right, take care.
Bye-bye.
Bye.